Good day, everybody. Good afternoon, good morning, from wherever you are joining us from. We are about to commence the program, the Day 2 program for the bootcamp. I'm sure we all learned one or two things from the class yesterday, and we must have some questions in our mind that we are willing to ask. Today's session is total practical, and I'm sure we are all with our junior software or our software tools. So yes, while we wait for others to join,
Okay, I think she's having a network challenge. So
thank you all for joining.
Okay, go ahead.
All right. Thank you all for joining. Please, let me see where you're joining us from in the chat box so we can know you are with us. Thank you. Okay, thank you all for joining from Nigeria. Ari from Pakistan, Bahati from Uganda, Abubakar. I see you all. Thank you all for joining. I'm sure we all had a nice time and benefited from, in a way or the other, from the bootcamp session yesterday. It's such a great opportunity. Today, you will have opportunity to continue the class that started yesterday. The CEO of Genomark, Mr. Oluwaseyi Olawale, would be the one to take us through the bootcamp session again today. And like I said earlier, it would be practical, all practical, fully practical. So get your devices ready, your laptop, the junior software, the tools and softwares you will be needing today. And in about a few minutes time, the CEO of Genomark would address your questions. It's also going to put you through. So yes, while we wait for others to join, in 6 minutes time, you will do that. So from Cameroon, from Nigeria, I can see a lot of people saying they've not gotten their junior software. On the group, there's a link to click on to make more inquiries. Please do that. I mean, before the class starts, click on the link. It will take you to a chat. Then you have a chat with somebody, and it would put you through how to get the junior software. Thank you. The message was sent yesterday, so click on it and we'll sort it out. Thank you all for joining. From Sierra Leone, from India, from Imo State. Thank you all from Malawi. You're all welcome. I'm trying to wait for others to join. And in about 5 minutes, the CEO of Genomark Holdings, Mr. Olawale Oluwase, will be taking over. Yes, presenting a slide already. All right. Thank you.
All right, so let's continue because we can't keep waiting for everybody to join before we start. So whoever did not start with us will definitely go through the recorded session on YouTube, right? So I want to be sure that you all have been able to go through previous class, right? You've been able to go through previous class, which was yesterday, and we'll be taking it over from there to address quite a number of different practical applications that we have here on Genius, and also with the use of other software tools or web tools that we're going to be using in the course of this bootcamp. All right. So yesterday we did hands-on internet tool for bioinformatics analysis, where we work with BLAST on NCBI database. So we worked on hands-on retrieving and handling NGS data retrieval from NCBI as a public database. So we also have hands-on Genius software. So I believe that quite a number of different people have been able to get either the free trial or the paid version of the lifetime activation key of accessing Genius software. And I'm going to open my Genius software right now because that's where we're going to start from. We're going to take it from data import, blasting, basic sequence analysis, so on and so forth, right? So, and in the course of this bootcamp, we're going to be addressing designing effective primers for PCR and sequencing projects. We also work on performing in silico cloning experiments, identification and analysis of motifs and repeat within DNA sequences, and also try to look at how you can identify the functions of these DNA sequences. Also look at the predicting open reading frames in genomic sequence, and also look at their functions of those open reading frame we've been able to predict or discover in a genomic sequence. Also, we have utilizing species finder and P2RP for species identification and regulatory protein analysis. Meanwhile, apart from this, we'll be looking at how to perform DNA hybridization. I just hope the time we have is enough to cover all of this. If not, probably uh, it may be probably in our next program, right? In our next program, we could maybe cover whatever we could not cover today, but I'll try my possible best for us to cover quite a number of different and some practical sessions as far as today's is concerned. So please follow us accordingly while we explore all these practical sessions. Just follow me accordingly, okay? So right now, like I said, we are starting with hands-on Geno software. So for those of you who already have Geno software, either free trial or paid version, and those who have it after this session, just in case you don't have the Geno software, you can still have the Geno software again after this session. So I'm going to open my Geno software now. So this is my Geno software here. So I can come here actually and search for my Geno software in case I can't, it's not on my tab. So I will just search from the home icon, right? I just search and this is my Geno software. I'm going to click it and my Geno software will open, right? So I'm launching my Geno software so that we can use it for different analysis as far as um, this this bootcamp is concerned. So I think my Geno software is opened. Okay, let me click continue. So I'm opening my Geno software now, and afterwards I'm going to take you on hands-on Geno software. We're going to look at how to import your data. Remember, we already retrieved some data set yesterday in yesterday's class. So today we're going to work on importing the data. I'm going to show you how to do blast also on Geno software. If you don't want to do blast on NCBI database as a web tool, there are other web tools actually out there that you can use for different kinds of bioinformatics analysis. They will also have basic sequence analysis that I'm going to show you based on the theoretical part that we were able to cover yesterday. I'm going to show you the practical application of the basic DNA sequence analysis covering the basic molecular dogma, where we look at how we can predict the complementary strand of the DNA sequence down to um, getting the codons and translating it so that we can get the amino acid that aggregates to form a protein. Okay. I hope you all are following and the class is becoming interesting. Okay, I'm opening my Geno software now. It's open. So this is my Geno software, as you can see on the screen. And right here on this part, these are where we create new folder and stores folder. And whatever that is in the folder that we click, that is what you see here. And whatever you click here, that is what you see on this interface, right? On this particular interface, and whatever the other tools and parameters you can actually touch on or use here around this area to be able to uh, decipher your DNA sequence and be able to elucidate it to draw insights based on whatever you are trying to look for or whatever you discover in the course of your analysis. So right on top here, we see the back blast agents, align and assemble. We see the three primers, cloning, and other kinds of analysis. There are also some kind of options we can click here to be able to pick some data, some options. All right, to be able to click or use some tool sets and some options that are available to carry out specific analysis or to do certain prediction. And I told you yesterday that Geno software actually is an embodiment of different bioinformatics tool sets that can be used to do different kind of analysis, right? That was what I told you yesterday. There are different kind of analysis from basic to advanced level that you can actually use bioinformatics for. I think that's one of the reasons why some bioinformatics companies are actually looking forward to be able to use bioinformatics at a whole different level, using genome software for various of different analysis because of the capabilities it can provide. Are you all following? Are you all following? Can you just quickly reply at the chat room, just for me to be sure you are all following? Just reply with a yes. All right, you can just reply with a yes. Okay. All right. So now let's start by looking at the data set we downloaded yesterday. So yesterday we downloaded the gene bank data set of a gene and also the FASTA file. Same, we downloaded the gene bank of the whole genome sequence of Acinetobacter baumannii, and as well the FASTA file of the whole genome of Acinetobacter baumannii. So I'm going to take it to installation now. I'm going to install it, or import it rather. All right, I'm going to import it. So this is local. So the first thing like you have to do right now is click local. This is Genome software. You click local, just like what I just did now. I've clicked local. All right, I've clicked local. So I go to file to create new folder. So it means whenever you come to Genome software, you want to begin to explore the data set, you want to import data set from the system. The first thing you have to do is what? You do what? You import your data. And how to do that is you click local, go to file and click new folder. Do you understand? And click new folder. Please, if your screen is not clear, just click the setting, click your screen, the video you're watching. You see the setting icon, click it to pick the quality, the highest quality of the video you want to watch so that you can see everything more clearly. All right. I repeat, this is Genome software. I'm going to import my data, and that's what you should do. The data set we imported yesterday, you would have probably imported or downloaded or retrieved data set from NCBI, either belonging to microbial, plant, animal, or human. Whichever one you try to do, just follow this next step to be able to see how we can elucidate and decipher our DNA sequence to draw specific genetic sequence analysis. All right. So now this is local, right? Like I said, then you go to file, you click new folder. So here I want to create a new folder. Now I'm going to rename this folder, right? Since I've just clicked new folder. So I'm creating a new folder and I have to rename it. So I'm going to rename it as Bootcamp tutorial, right? You can rename it as maybe basic sequence analysis, right? Whichever one you want to use. So let's just use basic sequence analysis. And I click it right now under local, you can see basic sequence analysis, right? So you see basic sequence analysis, which I just click now, basic sequence analysis. So I've clicked it. So right here, you see that there's no folder, there's no data rather, there's nothing at all. So we need to import the data set we downloaded yesterday from NCBI database. So to do that, I make sure that the file I just created is being clicked upon and highlighted like you are seeing. Then you go back to file, then scroll down to imports from file. Can you see import from file? Now, the moment you click import from file, it's going to take you to the folder, the system folder, right? So where you're going to look for downloads, all right? So currently I'm on download. So probably let me just look for, okay, I see gene bank, I see. So probably let me just open a new folder. Let me create a new folder. Let me just create a new folder and call it Bootcamp. Okay, I'm calling it Bootcamp. I just want to like make it easier for me to find my data set on Genome software. So I've actually cut and moved, or probably just moved my data set into the folder I just created now, which is Bootcamp. So I'm going to look for, let me refresh, and then I look, okay, this is it. Can you see this is the folder? I'm going to double click it. All right, so these are my samples or data sets. So I'm going to press Control A to pick all of the data sets and then click import. So I just click import. So it's importing all of my data sets. You can try to do one-on-one, you can import everything at the same time, right? And that is what I'm doing now. I'm importing the data set into Genus software. So it's asking me, do I want to keep sequence separate or create sequence list? Of course, I want to keep sequence separate because those sequences are separate sequences. All right. So I'm going to keep sequence separate. So I'm going to do again for the other one. So I should have 4 sequences, okay? 4 samples or 4 data sets in the folder, 4 data sets, right? Same here as well, right? So my Geno software is not responding. Let me just exercise patience. Okay, I think it's responding now. So here are my sequences. All right. Here are my sequences. Now I'm going to start exploring them one after the other. So, but you can just start by looking at uh, the description or the sequence length. So the sequence length will tell you that, oh, what you imported are genes. These are genes. These are whole genome, right? Because of course, whole genome sequence cannot have a sequence length of just 1004. So it means the 1004 is the sequence length of the gene. And the 3 million here you see are the sequence lengths of the whole genome sequence of Acinetobacter baumannii, right? So the moment I go ahead to click the sequence, so I'm going to know if it is the gene bank of the FASTA file. So here I'm seeing, I click one here, and then I'm clicking another, right? So now, what is the difference? How do you identify the gene bank file? How do you identify the gene bank file? So when I click the first one, I see this result. So this is telling me that this is the gene bank file because I'm seeing this annotation result. And what annotation I'm seeing here is the CDS, which means coding sequence. So it means in this gene, this particular yellow color you are seeing here, it is the annotation Hello, everyone. Please, can you hear me? I lost you for a moment. I think it was my network. Apologies for that. I just want to be sure you can hear me now. Please, can you reply with a yes if you can hear me audibly? All right. Can you hear me audibly now? Okay, okay. I can see you all can hear me now. So I'm back now. So let's continue. So as I was saying, right? As I was saying, um, the sequence I just clicked now is the gene bank file of the data set I imported. All right, for the gene, which is the gene bank file. So the CDS you see here, all right, is the what? Coding region of the gene sequence. Are you with me? Is the coding sequence of the gene. Do you understand my point? So the coding region. So this is the entire sequence. When I zoom in, you see one to what? 12104. But when I zoom out, the coding region begins from where? But when we put it here, we discover that the interval of this coding sequence is from 85 to 906. So every other regions are just non-coding region, and they are basically called introns. Okay, so the exons of this gene sequence is what we are seeing here, encoded by the yellow color, and it covers the region from the interval of 85 to 906. Is that clear? To 906. All right. So that's it basically. And then we have the translational data, or the translation sequence of that particular coding region. All right, which encodes the protein that is responsible for the biological function and processes of this particular gene. So what we see here, which is the FASTA file, does not contain the coding sequence, and that's just the difference. Are you with me? That's the difference. So now, if you are to use the sequence, the gene bank sequence of the gene of OXA23 gene responsible for the antibiotic resistance of this, I mean, for antimicrobial resistance to this particular antibiotics, which is class D beta lactamase, right? No, no, I don't think this was the I don't think this was the gene. I mean, this is actually the product, but it's resisting a particular antibiotics which we knew. I think yeah, it should be D beta lactase, right? So now, how do you do some other kind of basic sequence analysis? Is from getting the complementary strand of the data set. Can you see the complementary strand of the data set? Or we can use this to predict the translational region. So basically, the translational or the translated region, or the protein region, right, would definitely start from where the coding region begins. So every other regions that is not a coding sequence would definitely not give any translational translated protein sequence, right? So we will not see that. So basically, what we're going to see, right, would be the translational region starting from the regions that actually code for a functional protein. So the reason why this does not code for any functional protein even after translating is actually because the region are introns, they are not exons. So here we are seeing the exons part of the work of the sequence, and these are the kind of analytical results that you are going to be getting by the time you begin to study and elucidate, to decipher, or probably carry out analysis, basic sequence analysis on the data set. All right. So that's it basically. You can come here and click statistics here. Statistics tells you the entire details of the sequence you downloaded. Look at the length, 104, which is the sequence length, right? And then these are the frequencies of how much or how many times each of the um, nucleotide sequences, the 4 nucleotide sequences exist in the entire DNA sequence. So here we have the ACGT. The A is 330 and it contains 32.9% of the entire DNA sequence of this particular gene. The C is 158, which is 15.7% of the entire what? DNA sequence. The T is 308, which is 30.8%. And then the GC content is the combination of the C and the G. Okay, you see the GC content? The GC here, it means GC content, which is the combination of the C and the G number together, giving us a total of 36.4%. Are you with me? I believe some of you are asking a question. Please, what's your question? If in whole genome there is any other gene present, do this software highlight that gene too? Yes, I'm going to show you how. I'm going to show you how you can look into other genes present in the entire whole genome sequence data as well. And we can actually look for this OXA23 gene also in the whole genome sequence, right? We can actually give it a try if it is present in that particular sequence. It might not be present, it might be present, it depends. Okay, so that's it. We have here the rough TM, which is the rough temperature that is needed to break the complementary strand into one. The molecular weight, which is single-stranded DNA molecular weight or double-stranded DNA molecular weight. Okay, so it's just double of the single-stranded DNA sequence. Then these are the composition of the amino acid present in the translation, in the translated region, okay, of this particular DNA sequence, which is the frequency here, the A, which is 15. That means there's the A amino acid, right? It's like 5.5% of the entire DNA, protein sequence, encoded protein sequence from the DNA sequence exon region, okay? Is that very clear? So these are the codons. These are all statistics giving you a kind of basic understanding to the genetics basic sequence analysis of the data. So if you come here, you look at the annotations. These are the annotations present in the gene bank of this data. The CDS and the gene is just one. The DNA fold, you can wait on the DNA fold is made, but we can't wait. So you can just try that on your own end. This is the text view of the data set. And this is just the info, providing you the information of the data set. Now, I say that I want to show you how to do blast on Genius software. All you just have to do is click the sequence and come here, click blast. All right. So once you're done, leave everything at default, and then you click search. So the moment you do that, it's going to create a subfolder giving you a search result of the blast tool you are doing. Just make sure you are online before you do this. So it means that blasting is not necessarily only on NCBI, just like we did yesterday, but you can also do that on on Genius software here. All right, because it has a blast tool incorporated into the Genius system. So I believe you all can see we have 57.9 percentage complete of the search results we're doing on the blast sequence of the gene bank we have. All right. Yeah. Please, if the screen is not clear, click the screen. You see a certain icon. Click it to make sure that you pick the highest quality view you want, okay, based on YouTube recommendations. Thank you. Yes, if there's any other gene present in the whole genome, this particular software will also highlight it, and I'm going to show you how. All right, I'm going to show you how. So just please stay with me, follow me steadily. So this is the blast result, and this is the entire blast result we have for the data set. So right here you can see the, what's this? Let me just try to drag it. Okay, this is the bit score. Okay, so the bit score basically is talking about the score metrics of the blast heat that we have. Okay, what's happening? All right, so we have the grade, which is the percentage similarity or identity that we're able to carry out, right? So if you scroll down, you can see the 100% here. So this is the actual data we blasted. So, and then we have other percentage similarity based on blast. If you like, we can click it here. So can you see what I'm doing? So I try to click the grade here. I'm clicking the grade. So the grade here is telling me how I can actually rearrange the data to start from the very first down to the last based on the percentage similarities. All right, which is the grade. Then we have the each start, we have the each end. So here we're saying that the entire DNA sequence of this particular one was 098 DNA sequence, but there's actually 99% of the original sequence we have in this particular sequence. So, and then by the time you keep going, you just keep seeing other kind of data sets or results that you can actually use to report, maybe probably if you are doing a kind of blast based research and all. So, and you have the result in this subfolder, right? So that's how you do what's it called right now? That's how you do blast. And then you can do query centric view to look at the alignment view, which would definitely tell you variations across different sequences. You understand my point? Across different sequences, similarities and differences across differences. I mean sequences, because I told you blast tool has to do with uh, checking for the differences and similarities between two sequences, right? So when you check for differences and similarities and differences between two or more sequences, uh, that is what BLAST is. So basically, you use BLAST to check for similarities and differences, which gives you insight into the evolutionary state of those sequences. So with this query-centric view, is giving us a kind of a simple view of sequence alignment using the BLAST metric view to give us insight into percentage similarities and differences, which will in turn give us or enable us to draw specific conclusion on the evolutionary states of the sequences. And let me tell you something: the evolutionary state of the sequence is determined also by basically the similarities and differences, which has to do with mutation or variations. So because for two genetic sequences to be different, it means that there is difference in genetic sequence or nucleotide basis. Do you understand? There's differences in the nucleotide basis, and the nucleotide basis is the ACGT based on the sequence. So if sequence A at region 50 is has nucleotide C, and in sequence B at that same position has nucleotide T. So it means there's variation. Variation has occurred at that point. So, and that is how you get to discover using this point. So we have over 100 sequences here. As you can see, we have over 100 sequences here. And these sequences we see here, all right, the sequences we are seeing here will give us an overview to understand the state of the variation, looking at the similarities and differences between them. Now, if you look at these sequences from here to here, you discover that some things are missing. So it means there's a genetic differences in this
I think please kindly stay tuned while we try to resolve that.
Hello, please, can you hear me now? Hello, can you all hear me? Hello, can you all hear me? You can reply with a yes if you can hear me, please, so that I can continue. Apologies. I have a very bad network here. So the network is really bad in this area. Okay, so I can see some people are hearing me already. So I'm going to be sharing my screen. All right. So meanwhile, I'm trying to answer some questions while my screen is loading. Okay. So some CDS are uploaded with all sorts of sequences, and this might affect the quality of what we do. All sorts of sequencers. Well, it depends. Some sequences are uploaded with all sorts of sequencers. Well, I wouldn't say, because before you have CDS, it means you must have done certain pre-processing. All right. So maybe like quality control on your data before you do some annotation. So when you annotate and you get some annotation, I believe you shouldn't have some other errors. All right. Because before you get there, you must have processed your data set. Right. You must have processed your data set, and that is very, very important. Okay. So I'm still trying to share my screen. Please, if you want to use the free trial, you can download the latest version. But if you are using the lifetime activation key, please work with the version we talked about. I'm talking about those who want to get Geno software, okay? Those who are getting the Geno software. All right, so that is it on this blast. So, and that's it on talking about the basic sequence analysis. Meanwhile, there's something I want to show you. All right. So after query centric view, we have the distances. So this distances here tells you, let me change this from numbers only to each maps and numbers. I'm changing to each maps and number here. Here is telling you about, it's more or less like DNA gene hybridization, telling you about the similarities and sequences between two samples. All right. So it's telling you how related a particular sequence is to another. It's like comparing. Do you understand? The percentage similarities or differences present within a sample and another. So this is like a comparison. You can see sequences arranged on the edge here, and you can see other sequences arranged at the edge here. So when you compare this with this, it's actually the same sequence. So this sign means identical. And when you compare this with this, definitely you will not say identical because this sequence is different from this one. So that's how you compare. You compare this with this, it's identical because it's the same sequence. You compare this to this, it's 98, 98.400%. You see that? 98.4. I can reduce the decimal point. I can reduce the decimal point to 0. So you just give us a definite measure. I can increase the decimal points to 2. So we have 98.4%. I can increase it to 2, like 98.40%. Are you getting the point? So it depends on the number of decimals you are picking here. You understand? And here we have percentage identity. That is the percentage of bases or residues which are identical. We have percentage of number of bases and residues which are identical. So I could decide to pick this, right? No, it's telling me total number of sequences that are identical. So when I compare with this, it's identical. When I compare this with this, I'm saying it's telling me that comparing this sequence length and this sequence length, there's only 940 sequence length identical. That's percentage identities of nucleotide bases between two or more sequences. Are you getting the point? Then we have percentage difference. So what's the percentage difference? So it's telling me that when I compare this to this, it's only 16% differences. You understand? So it depends on whatever you want to pick to look at the evolutionary emergence that has occurred between two sequences. That is evolution at gene level. Do you understand? Or at genetic level. And that is how you're able to get some of this insight when you compare sequences with another. So to compare others, you just go like that, and then you come here, you compare with this, you have this, you compare with this, you have identical, you compare with this, you have 100%, and so on and so forth. So that's how you compare. You compare this to this to this to like this. You know, you keep comparing in matrix format until you are able to compare everything together. All right. So that's key, and that's all about the gene. So here, when we go to whole genome level, we actually see a whole lot of different sequences present, right? So this is whole genome sequence, and this particular view you see here is in circular view. So it means the sequences are in circular view. The whole genome sequences are in circular view, isn't it? Can you see? We can see some circular view of the entire sequences shown here, right? Circular view of entire sequences. So I could decide to remove the circular view, and this circular view you see also have other annotation view. So but to remove the circular view, I just need to come to sequence and click circular view so that I can remove it. Right now I've removed the circular view, and I just need to zoom in. So right now I'm zooming in to see every annotated feature, because the whole genome sequence I'm seeing here is the gene bank file of the whole genome sequence of Acinetobacter baumannii. Do you understand? And it contains 3,752 CDS, 3,856 gene, 3 ncRNA, 6 regulatory sequence, 18 RNA, 1 source, 1 tmRNA, 76 tRNA. Can you see that? So these are the annotated view of the sequences, and I could decide to look for a specific gene I'm looking for right here. How do I do that? I can actually view